Welcome again for the student in College of Medicine and College of Pharmacy whom join us in the series of lectures in the physiology of cardiovascular systems. The summary of the previous lectures. The heart is formed from 4 chambers, 2 atria, an interatrial septum, and 2 ventricles with interventricular septum, and the 2 systems, pulmonary who carry deoxygenated blood and systemic who carry oxygenated blood. And as we discussed, there is a 4 valve, 2 atrioventricular valves and 2 semilunar valves. The atrioventricular is the bicuspid mitral and tricuspid valve. Semilunar valve is aorta and pulmonary. There is coronary circulation composed of right and left coronary arteries. And layers of the heart is endocardium innermost, myocardium is the bulk of heart, and pericardium the outermost. We will discuss the physiology of the heart under 3 levels or 3 sessions. At cellular level, we will discuss action potential and automaticity. And at the muscular isolated muscle fiber, we will discuss the mechanism of the contractions event of the cardiac muscles and excitation contraction coupling and calcium induced calcium release by these mechanisms. And at the organ level, isolated heart, and we will with the integration of the whole cardiovascular systems, and we will discuss the cardiac cycle. The heart as organ is innervated by 2 ways, by 2 systems. By the sympathetic system, from sympathetic chain from anterior whole cell and the dorsal ganglia and the anterior whole cell and ganglia, and they stimulate the heart. Stimulation of sympathetic is the cardioaccelerotory center. It means that they make the heart rapid beating. Heart is inhibited by parasympathetic, which is by vagus nerve, the nucleus in the dorsal motor nucleus of the vagus nerves and down to the innervate the heart. So there is 2, cardioaccelerotory and cardioinhibitory. Cardioaccelerotory by sympathetic stimulation, cardioinhibitory by parasympathetic stimulation. Conduction system of the heart, as we discussed in the first lecture, is the specialized system for the conduction of the electrical activities of the heart, which is form about 1% of total structure of the heart. It forms from SA node, sinoatrial node, which is called natural pacemakers. This node in the right atrium near the superior vena cava, and the impulse transmitted by internodal and the atrial conduction systems. Then to the AV node, atrioventricular node. There is delay, a fraction of a second there, and transmitted to the bundle of His at the upper septums, and from bundle of His, there is the bifurcation of atrioventricular bundle branch. This is left bundle branch composed to anterior and posterior segment, while this is right bundle branch block, ending with the Purkinje fibers at the end of the conductive system. So impulse transmitted from SA node to the atrioventricular node, bundle of His, bundle branch down to the Purkinje fibers. This again, how to imagine, this is the conduction system, SA node, internodal connection to the AV nodal system, from AV node bundle of His to the left bundle branch block, right bundle branch block, ending with the Purkinje fibers. It consists of pacemaker cell and conduction pathway. Pacemaker cell is a natural pacemaker, is the SA node, which is dominate the heart, which is discharged spontaneously, and the impulse from SI node down to the by conduction system to the other part. And the electrical activity, this coordinate contraction of atria and ventricle. There is two functions, electrical activity and the mechanical contractions. The electrical activity is that preceded that the ventricular contractions and atrial contraction and coordinated and really end before that the contraction is ended. So another review for that the SI node, that the upper part of right atrium and impulse discharge in right and left by internodal connection to the AV node. There is some delay there of the impulse and the bundle office and bundle branch block Purkinje fibers. So the impulse conduction normally to start from the SI node, sinoatrial node, which is cardiac pacemaker, discharge to the internodal connection, interatrial conduction system to the AV nodes, and from AV node bundle office and the right and left bundle to the Purkinje fibers. And the impulse really normally in adult between 60 to 100 beat per minute, normally. Now come to the electrical activity at the cellular level, physiology of the cardiac muscle at the cellular level. Well, look, there is a channel. It's a sodium channel and potassium channel and calcium channel, 3 channels. As this channel and the voltage gated, it means that the ion it passes from the high voltage to low voltage. That's voltage gated. And that the resting membrane really between the -90 to -80. This is for the myocyte fibers, not SI node, and the myocyte. And that the phase 0 of the resting membrane potential and resting membrane voltage is the increasing from the minus to the really near the -70, and at this level that the myocyte, it will be spontaneously and it will be discharged the signal. Look how discharge is, and the sodium channel is open and the fast sodium channel is passed. This is that the phase depolarization. Phase 0 is the depolarization of the myocyte. Due to that, the influx of sodium ion with a positive charge inside the myocyte. After that, there is a phase 1, which is early phase. Look, to the potassium channel is open, and potassium slowly efflux out the cell, and this is early phase of repolarizations. While the next phase is coming is the slow phase, due to the slow sodium channel, L sodium channel. We have L and T. This is a slow L. And look to that, the slow influx of the calcium from outside, and they take a long time, which is the about 100, even 150 fraction of millisecond. And this is important for the cardiac muscle plateau due to the influx of calcium channel inside the cell. This is give a time for the heart to be filled during diastole. This is not present if you study the striated muscle. There is no plateau in the striated muscle, only in the cardiac muscle. This plateau is important phase for the give more time for the filling of the atria during end part of diastole, to fill the blood, to fill the ventricle atria with the blood, to prepare it for the ejection. And the third phase is the rapid and I mean more potassium outside. This is the phase of, and the final phase of the repolarization. So this is depolarization. This is the phase of the repolarization. The last phase, most of sodium will be out and potassium will be out. This is energy dependent due to the adenosine triphosphatase. This is high energy particles or molecules. So most of sodium will be out in its normal resting state. Most of potassium will be, and the calcium will be deal with the inside the cells by endoplasmic reticulum. And so the net, this is the sodium out and potassium in and resting phase. And this is what called action potential in a single cell. And it is transmitted from one cell to the whole heart to synchronize the atrial and ventricular contractions. Within 0 phase, 1 phase, 2nd phase, 3rd phase, 4th phase, and equal 5 phases of the depolarization and repolarization. During depolarization, the ventricle is contracted. During repolarization, the ventricle is relaxed. And while in SA node is quite different. The SA node have no, it depends on the calcium and potassium only, and have no rapid, I mean fast sodium channel, because that the sinus nodes is not have resting. It is continuously discharging. This is what called automaticities. So that the membrane, I mean at the node at the minus 90, at the minus, near the minus 60, and it will be discharged, and there is no plateau and discharging again. It is phase 4 only. This is for SI node. So this is action potential, depolarization, repolarizations with a plateau. This is the main phases, early repolarization and late repolarization, and phase 4 is the resting phase. Am I clear? Okay. So autorhythmicity cells, which is initiate action potential, mean SI node, which is the pacemaker of the heart, have unstable resting potential, not stable. If we look to that, this is that all the graph, previous graph. This is resting. It's not unstable at the discharging threshold always. So it is and then discharge again. Pacemaker potential. This is what called unstable resting called pacemaker potential. Use calcium influx inside rather than sodium for the rising phase of action potential, and potassium it will be out. The SA node generate impulse about 75 beats per minute. That is say 60, between 60 to 100 beats per minute in adult. Atrioventricular node delay the impulse approximately 0.20 second, about 100 millisecond. This is for the plateau, for more time of diastole to fill the heart. This is depends on the action potential of the pacemaker action potential on the normally potassium in, so calcium out. But during the contraction or discharging, calcium in and potassium will be out, and no resting potential from the minus 60, the threshold of discharge, while in the myocyte is the minus 80 to minus 90. So impulse passes from the atria to ventricle via atrioventricular bundle branch of his AV node, split into the two pathways into the interventricular septum upper part bundle branch, right and left. Bundle branch carry the impulse toward the apex of the heart. Purkinje fibers carry impulse to the heart apex and ventricular wall. Look to this shaded diagram. This is SI node discharging from impulse there, from SI node, and the impulse from discharging to the both atria, left and right, and sometimes and they take a moment to delay in the AV node. From the bundle branch, it's contract the ventricle. We will see that the depolarization of atria give the wave in ECG called P wave. We'll discuss that in details in the coming lecture. While the ventricular contractions, I mean depolarization, give QRS complex. So that the depolarization of atria give P wave, not contraction, but depolarization, which preceded the contraction of atria. Then depolarization of ventricle give QRS complex, which preceded the contractions. So this electrical activity we will find recorded in the surface of the body by machine called electrocardiogram, and the paper called electrocardiography. We have about 5 or 6 lectures in the ECG in the coming lectures. The P wave corresponds to depolarization of SI node. QRS complex corresponds to the ventricular depolarization, not contraction. T wave corresponds to the ventricular repolarization. Atrial repolarization record is masked by larger QRS complex. Repolarization of atria not seen in the surface ECG because the repolarization of ventricle with the bulk muscles and that the T wave is that the mask that the QRS complex by QRS complex of atrial repolarizations. Why the ECG is called by PQRS? It's arbitrary. At the time of recording or the nomination of the ECG, they put PQRS and the T wave. Have no other meaning, only putting there to name the wave. This is the atrial depolarization called P wave, while the QRS, the ventricular depolarization, what called the QRS complex, and T wave is the ventricular repolarization. And we will discuss this in details with this wave and interval and how to read ECG in the future. Again, this is important. This P wave is the ventricular, atrial depolarization with at the end of this early diastole at the end of diastole to fill the ventricles. And when you start the ventricle contract asystole, give a QRS complex. And T wave is the early diastole by relaxation of the heart by repolarization of the ventricle. While the repolarization of atria, it masked there. It's not seen in the surface ECG. So conduction, cardiac conduction of impulse was generated in the cells by action potential, by the potential gated channels, sodium, potassium, calcium. This is to make the heart contracted, to pump effectively large portion of cardiac muscle receive an action potential nearly simultaneously at the same time transmitted by junction gap. This junction gap is the easily and the very easy to pass this action potential from one cell to other cell. Special cells that conduct action potential extremely rapidly are arranged in a pathway through the heart. Before mechanical contraction, there is electrical action and action potential travel quickly over each cell membrane and down into each cell and transmitted from one cell to the other cell by intercalated disc, which is the freely and easily pass the action potential electricity from one cell to another cell. Three physiological characteristics of specialized electrical cell. The nodal cell and Purkinje cell provide these synchronizations. So these three characteristics: automaticities, it means ability to initiate electrical impulse without stimulation. If you remember the skeletal muscle, you have to stimulate for the contractions, while here automaticity, the cardiac muscle have ability to discharge spontaneously. Excitability, other characteristic, ability to respond to electrical impulse. Conductivity, ability to transmit an electrical impulse from one cell to another. So these three characteristics: automaticity, excitabilities, and conductivities. So in summary, sinoatrial node fires at rate of 60 to 100 at the mean 75 beats per minute. Internodal pathway, atrioventricular node fires at the 40 to 60 beats per minute. And atrioventricular of Bandelofis and ventricular tissues fire at 20 to 40. We see that the impulse firing from up down is that the fastest up, 60 to 100, slow in that the atrioventricular, more slow in ventricle. That is why the SI node is dominate of the other part, which is natural pacemaker, dominant pacemaker of the heart. Right and left bundle branch, then Purkinje fibers. And this is another view, the phases of this resting membrane, as we see, minus 90 during depolarization near the 30, plus 30 reach. And the sodium will be pushed in, and calcium pushed in during plateau, and potassium, it will be pushed out, and the sodium will be pushed out, and sodium efflux. And this is the phase 0 and phase 1 early repolarization, plateau phase, and the phase of final repolarization with the resting phase. Look to the resting, how the positivity in outside due to the sodium ion, and negativity dominant inside due to the negative ion there, less positive ion intracellular. During depolarization, more positive, more sodium is coming due to the fast sodium channel inside, while during repolarization and return newly to the resting phase. Now to this is I think enough at the cellular level in the physiology of the cardiac muscle. Now to the isolated muscle fibers. I mean to study the physiology of the heart at the level of the muscle fibers. What the muscle fibers of the heart characteristics? Cardiac muscle is striated like skeletal muscle, short, not long fibers like skeletal muscle, fat, branched. What mean by branch? We will see it's connected and branched again, and interconnected by disc, intercalated disc. Connective tissue endomyosine acts as the both tendon and insertions in the myocardial, in the muscle of the myocardium. Intercalated disc anchor cardiac and connect cardiac cell together and allow free passage of ion. That is why the action potential is the spread rapidly and to coordinate the chambers of the heart to be contracted simultaneously and in a synchronized fashion. Heart muscle behave as a functional syncytium. Look to this, the characteristic of anatomy of heart muscle. It is short, branched and joined like lattice work, and this is in nucleus, one nucleus only, and this is the intercalated disc, as we say. It's not present in the striated muscles. And this is intercalated disc, which is make that the electrical activity pass easily from one cell to another. So it is a branch are connected, branch and connected. And this is that as a cell of a mitochondria, and there is the picture here, look, intercalated disc and sacrolemma outside. This is sarcomere. What I mean by sarcomere is a contractile unit of the heart. We'll study it in a more details. This is intercalated disc, and that the faint part is called I-band. Well, I-band is that the thick looking of that. This is sarcomere. Take a muscle fiber. Please, this is important. It's coming in exam more than one time. The characteristics of the cardiac muscle, striated like skeletal muscle. Short branch cell, as we see, it's short, divided and connected. Uninucleated, one nucleus. Intercalated disc, which is not present in the skeletal muscle. T-tubular and over that disc. We will see what I mean by that disc. This is a mitochondria and the nucleus, and this is the fibers and the contractile fibers and contractile unit, what called sarcomere. This is the fibers of the myofibrils, what's called, and that they are surrounded by calcium-containing sarcoplasmic reticulum. And this is the thin, I will discuss the thick and thin filament and the sliding theory like the skeletal muscle if we study it. So come to the contractile unit, is a sarcomere. Have a thin filament and the thick filament. Thin filament is called actin, thick filament called myosin. And the thick filament resting on a line in middle, what called ame line, while thin filament resting on a line, what called Z line. This is a Z line, and this is ame line. Z line for the thin filament, ame line resting of thick filament. There is structure important in a thin filament and a myosin. There is two proteins important, troponin C and the tropomyosin, or troponin complex, tropomyosin. Tropomyosin, it's attached to these heads of and the thin filament, while troponin is activated by the calcium, which coming from the outside the cell, while the myosin thick filament have a head of myosin and resting in the M band, or M line. And we will discuss how these actin, myosin, thin and thick filament important in a sliding theory of contraction and relaxation of this sarcomere. This sarcomere is a contractile unit of the myofibril of the heart, which form from actin and myosin. And this is the sarcomere model. I look to this is a thin filament and thick filament. Thick filament on the M line and thin filament, and actin, myosin, myosin there and actin in this is the Z line and this is M line, and during contraction, relaxation, and each one is sliding over another. This is the head of the myosin thick filament. We will see how it's important, this head, for due to the sliding theory. So again, in more details, the thick filament myosin resting on M line, while thin filament, which on the Z line, actin have troponin C and the tropomyosin proteins, and while the myosin thick filament have the head of this myosin. As we said, this is A band and light, and the I band, this is A band. What the mechanism and contraction event of cardiac muscle fibers? So cardiac muscle fibers contract via excitation contraction coupling using a mechanism unique to cardiac muscle called calcium-induced calcium release. So contraction, excitation contraction coupling in the myocardium, striated muscle, some smooth muscle, while calcium-induced calcium release is unique for the myocardium. Excitation contraction coupling describe what I mean by that process of converting electrical stimulus, which is action potential, into the mechanical response, I mean muscle contraction. While calcium-induced calcium release involve a mechanism in which involve the conduction of calcium ion into the cardiomyocyte, triggering further release of ion into the cytoplasm. What I mean by this? During study of the striated muscle and skeletal muscle, we find the source of calcium from endoplasmic reticulum only. While in the cardiac muscle, there is two sources, one from by calcium channel from outside. This calcium which enter inside, which is the activated troponin, which is released at tropomyosin, and this is also lead to the activation of endoplasmic reticulum to release more calcium. So it is called calcium-induced what release calcium, more calcium from endoplasmic reticulum. So calcium prolong the duration of muscle cell depolarization. If you remember in a plateau, as we said, there is more time due to the calcium. This is not present in the skeletal muscle. Before the repolarization occurs, contraction in a cardiac muscle occurs due to the binding of myosin head in a thick filament to the adenosine triphosphatase, which is energy molecule, which is split to the adenosine monophosphate and the phosphate molecule, which is then pulls the actin filament to the centers of sarcomere by the mechanical force of contraction. Please remember, this is during entrance of the calcium. The calcium stimulated troponin. This troponin is the dislodged tropomyosin, our troponin complex protein. This receptor is free, so the head of myosin is coming attack and pull the thin filament inside from. So sliding it inside, this is during contractions. While during relaxation, the endoplasmic reticulum, which is release more calcium, what's called calcium-induced release of calcium from endoplasmic reticulum by itself dealing with this excess sodium, calcium, so that the tropomyosin return back and that the relaxation and the head of myosin is released at the actin, so it is lead to the relaxation. So it is become short during contraction, become long during relaxation. I think this is clear. What I mean by this term, excitation contraction coupling, as we said, the physiological process of converting an electrical stimulus to the mechanical action, mechanical response to the contraction. As we said, the heart is the pump machine, mechanical machine. Calcium-induced calcium release, a process whereby calcium can trigger release of more calcium from endoplasmic, sarcoplasmic reticulum. This is in a cardiac muscle only, while in the skeletal muscles, and in the skeletal muscle, and this is the only calcium source from the sarcoplasmic reticulum. Cardiomyocytes are capable of coordinate contraction, control through the gap junction, as we said, of intercalated disc. The gap junction spread action potential to support synchronized and the simultaneous contraction of both atria and ventricle as the one syncytium, one unit. In a cardiac skeletal and some smooth muscle tissue, contraction coupling through a phenomenon called contraction excitation coupling also present. But that the calcium induced only release of calcium only in myocyte. Describe in the process of converting electrical stimulus from the neurons into the mechanical response that facilitate muscle movement. Action potential are the electrical stimulus that elicit the mechanical response. That is why before the mechanical response. And the cardiac muscle, and the ECC, is dependent on a phenomenon called calcium-induced calcium release, which involves influx of calcium ion into the cell from outside, trigger further release of ion into the cytoplasm from the sacroplasmic reticulum. The mechanism for the calcium-induced release is a receptor within the cardiomyocyte that bind to calcium ion when calcium ion channels are open during depolarization, releasing more calcium into the cell. Similarly to the skeletal muscle, the influx of sodium ion causes an initial depolarization. However, the cardiac muscle, the influx of calcium ion sustains depolarization. That's what's called a plateau, so that they last longer, create at the plateau phase in which the cell charge stays slightly positive, depolarized, give more time for filling of the heart. This briefly, as we said, is about 100 milliseconds before it becomes more negative, as it is repolarized due to the potassium influx. Skeletal muscle, by contrast, repolarized immediately. There is no plateau phase. And I mean depolarization, repolarization. The actual mechanical contraction response in cardiac muscle occur via sliding filament theory, a model of contractions. And the sliding filament, I mean sliding actin over myosin filament in the sarcomere, the contractile unit of the heart, and this is myosin filament slide along actin filament to shorten or lengthen the muscle, shorten during contractions and lengthen during relaxation. The pathway of the contraction can be described in the 5 steps. I mean muscle contraction, sliding, actin, myosin, sliding each one over others and to shorten the sarcomere, and it is during relaxation and it's lengthening the sarcomere. This is by 5 steps. First steps is action potential, as we say, electrical activity before the contractions, induced by pacemaker cell, as we study in a sinoatrial node. And then AV nodes is conducted the contractile cardiomyocyte to the gap junction, which is freely passed and easily passed to the whole cell. As the action potential travel between sarcomeres, it is activate the calcium channel in the T-tubule, resulting in the influx of the calcium ion into the cardiomyocyte. Third phase, calcium in the cytoplasm then bind to the cardiac troponin C. It's activated, which is move the troponin complex, or tropomyosin, which is cover the binding site of head of the thick, of the myosin thick head. So away from actin binding site. This removal of a troponin complex or tropomyosin protein frees this actin to the bound the head of myosin and initiate pulling of the short, thin filament inside. This is the initiate the contraction. Fourth phase is myosin head bind to the adenosine triphosphatase energy molecule and pulls the actin filament toward the center of sarcomere, contracting the muscles. While in the fifth stage, intracellular calcium is then removed by sarcoplasmic reticulum, dropping intracellular calcium concentration, returning troponin complex to its inhibiting positions, and the head of the thick filament is released, and the ending contraction as actin filament return back to their initial part, and this is what called relaxation of the muscle. So this is the actin and myosin. This is during contraction and during relaxations. The red is that the red filament is that the myosin, and which is on that the, and this is that the actin, and this is contraction relaxations of that the. So heart muscle, cardiac muscle fibers undergo coordinated contraction via calcium-induced calcium release conduct through that the intercalated disc, is stimulated by nerves and it's self-excitable. As we said, automaticity. Contract as syncytium as one unit, has a long, about 250 millisecond absolute refractory period, in AV node to delay the impulse. Cardiac muscle contract in a similar to the skeletal muscle with some exception as we discussed. Thank you for active participation. See you soon.